Good morning, everybody. So today we are going to begin our course for your NEET exam, and for this I'll be taking up the zoology portion. And in zoology you must have seen the first topic, that is animal kingdom. So we will begin with this animal kingdom, and while you are studying the topic you can keep raising your doubts. You can ask those doubts through the chat. I'll be getting those doubts and I'll try to clear as many as possible. Of course, all of the doubts will be cleared. Second thing is that you must have already prepared, because earlier the exam was on 3rd May. So you are, I feel, comfortably prepared for the topics. The only thing is that even though you are prepared, but there may be some lacuna here and there. So this is, through this course, all that lacunae will be filled. So try to make the best of it, ask your doubts and try to note down whatever is given to you here. So let us begin with our first topic, as we said, animal kingdom. Now, animal kingdom also is called metazoa. So this is also called metazoa, and metazoa, this term according to the 5 kingdom classification, what all organisms are included in animal kingdom. So first thing we can mention that these are multicellular eukaryotes. Okay, so these organisms which we study in animal kingdom, they are multicellular eukaryotes. That means their body is composed of many cells, and of course eukaryotes, you know that their nuclear material is enclosed in a membrane and they have membrane lined organelles as well, right? So this is first thing. Now multicellular eukaryotes, even the plants, the fungi are also there, but there is one more thing that makes them different, and that is these are all heterotrophic. Heterotrophic, that means they are the consumers. And one more thing that we can mention, they lack cell wall. Okay, so animal cells, of course, they are lacking cell wall, they don't have that. So these are 3 very important characteristics. Then one more characteristic that we can mention is that is animals have the ability to move from one place to the other, and that is locomotion. Now as soon as I write locomotion, some people immediately will have a doubt that you will say that sponges, they are sessile, they are attached to the substratum. Sea anemones in the cnidarians and many of the other cnidarians, they are also sessile, they don't move. So let me clear this point a little more, that locomotion at least at some stage of life. Even if that is true, then also we can take it as a characteristic feature of Animalia. And you will see in the sponges also they have a larval stage which is motile. Even in case of sea anemones, there too the larval stage is motile. So that way we can certainly take it as a identifying feature. Okay, so locomotion at some stage of life. So even if all through life they are not motile, but even if at some stage of life they have locomotion, we can certainly take it as an identifying feature. And last but not the least, the animals have the ability to sense their surrounding. They have some nerve structure. Now once again sponges are an exception because they don't have any nerve cell, right? So in sponges there is no nerve cell, fine, but that doesn't mean that they don't feel. There the division of labor hasn't occurred. All the cells of the body have the ability to sense their surroundings. So they are also very much animals, right? So here once again we can mention that ability to sense their surroundings. Right? So these are the most important features that I have mentioned over here for animal kingdom, right? And I don't think up till here anybody would have any problem. Now, taking out some brief classification, although this classification is slightly more than the NCERT, but anyway, few things you need to know. So first of all, you see metazoa, this is the name of the kingdom, right? So this metazoa, it can be divided into two subkingdoms. So here, one subkingdom is parazoa. Okay, so one subkingdom is parazoa, and in parazoa we study only one phylum, that is Porifera, right? Now in parazoa, which all group of animals we have taken, the basis of the classification, that these parazoans, they don't have any symmetry, they don't have any tissue. Okay, so we can just combine this here, no tissues, no germ layers, and no symmetry. So it's easy to remember this way, that what all these parazoans are lacking, that they are lacking symmetry, they are lacking germ layers, and they are also lacking in the symmetry, the tissues. They don't have any tissues, right? And in this, as we said, the only phylum that we study is phylum Porifera. Phylum Porifera. Okay, so the other subkingdom, I'll take it over there. Okay, so the other subkingdom, now this subkingdom is Eumetazoa. Eumetazoa. Now U term, you know, whenever we have the term U, that comes for which are true metazoans, right? Just a sec, this part of the board I think is not visible. I'll just shift a little. Okay, so we can carry on, right? So this Eumetazoans, in this there is presence of tissues, and some will be going higher than this, that is, we can mention organs, then some can go even higher, so that would be organ systems. Okay, so this will be in case of these Eumetazoans, presence of organs, organ systems, and of course that starts with the beginning of tissues. Okay, then apart from that, next thing we can mention germ layers. They do have germ layers. I'll just be mentioning what germ layers are so that it is crystal clear to you. So germ layers, where the number of germ layers can be either 2 or 3. Okay, so germ layers 2 or 3, these can be there. And last but not the least, of course, they do have a symmetry. Okay, but exceptions are always there, but mainly symmetry is present. And this symmetry, then we will be further studying that this symmetry can be either a radial symmetry or it can be a bilateral symmetry, right? So first thing that you see here is just the basic beginning we have taken, that when we talk about Animalia, that is animal kingdom, so animal kingdom is also called metazoa, and this metazoa has some characteristic features, which 5 of them I have listed over here. Okay, And then this metazoa is divided into two subkingdoms, and these subkingdoms are parazoa and the other is the eumetazoa, true metazoans, right? So parazoa is a blind-ended line because this was a offshoot, you can say, and then it ended blindly and it did not further lead to any other phyla arising from it. So it was a evolutionary dead end. While the other branch, starting from the original ancestor, you see, that gave rise to these eumetazoans, from which there has been several other branchings, and those have led to the other phyla which we will be studying. Okay, now in eumetazoans, what all phylum will be there? So here we will have phylum Coelenterata, or which is also called Cnidaria, Coelenterata or Cnidaria, and then there is after this Ctenophora, then Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and last but not the least, Chordata. Okay, so now we have a list of all the phyla which we are supposed to study in this chapter, and these phyla, as you can see, one is Porifera and the remaining are mentioned over here. So these many are to be studied. Okay, so here, but before we delve into these phyla, into the details of these phyla, we first take that how what is the basis on which we have classified this particular animal, like whichever animal the scientists they discover, then they have to categorize on the basis of certain features where it has to be put in one phylum. Okay, so suppose a new organism is discovered, so there would be some characteristic features on the basis of which the taxonomists, the systematists, they will decide that it should be put under maybe Arthropoda or maybe Annelida, whatever. So there has to be some basis, some way by which we can sort out, that is a way of sorting the various animal groups into different phyla, right? So here the first part of the chapter that we take is the basis of classification, and let me tell you, this is the most important part of the chapter, because maximum questions you will be able to solve from just this part of the topic. And then of course for examples and some special character which is with the particular phylum, that we will be doing with the type study, of course. But here, first thing, the most important part of the topic, that is the basis of classification. So let us begin with the basis of classification, the basis on which we are putting, we are sorting the animals into so many phyla. One of the bases we have already mentioned in short. You can see that whether the multicellular body is just having cells or the cells have been organized into tissues, organs or organ systems. And then the other one, that is whether they have germ layers or not, and if the germ layers are present, they could be 2 or 3, right? And of course the symmetry. Okay, so let us start with the basis of classification. So here, starting with the basis of classification. Okay, the basis of classification. Now you see the basis of classification. First we can start with the levels of organization. Levels of organization. Now levels of organization, you see, it is simply a hierarchy, that is going from simpler to complex. So this is a hierarchy, and as you know that the structural and functional unit of life, that is the cell. So the smallest unit of life is the cell. So that is the first hierarchy level. So we'll start from there. Okay. So as you know that the first thing, cellular level. Then these cells, they group together to form tissues. So next would be the tissue level. Then from tissues there will be formation of organs. So organ level, right? Organ level. And then finally when the organs group together to form organ systems, so this is organ system level. Okay. So in this case, first if we see cellular level. Now with cellular level there is only one phylum which has this level, that is phylum Porifera. So Porifera is the one phylum which is having just a body consisting of cells, nothing else. Okay, so the cells have not yet organized into tissues. So each cell has its some degree of independence possible within it. Okay, so that is the cellular level. Then cells, when they organize to form the tissues, so that would be the next level of organization, and this next level of organization is with phylum Coelenterata and Ctenophora. Coelenterata and Ctenophora, right? And then further, like for example tissues, you know that in the animals there are 4 different types of tissues, right? Epithelial tissue, muscular, nervous and the connective tissue. So when the cells having same origin, they group together so that they can carry out a particular function, they form a tissue, and this way here the tissue level has been achieved. Now if we go further, that at the organ level, like if you talk about stomach is an organ, right? And in this stomach you must have studied the structure in the histology of the gastrointestinal tract, where you have seen that when we take a section through any part of the alimentary canal, you will find that there are 4 different layers present, and those 4 different layers you will find there is epithelial tissue also present, there is connective tissue also present, muscular tissue, as well as number of neurons are also present within it, right? So you will see that all 4 tissues are combining together to form a organ, right? So here organ level, where the organ level has been reached, so that is the phylum Platyhelminthes. Platyhelminthes, which is flatworms, right? Tapeworm, it comes under Platyhelminthes. Then organs combine together to form organ system. Like in the digestive system, once again taking the example, there is esophagus, stomach, small, large intestine, all that are combined to form the organ system. So where this organ system has reached, that is starting from Aschelminthes with the phyla. I have already listed one, so that next time we can just put that from here to here. So here it is phylum Aschelminthes to chordata. Ashkelminthes to chordata. Okay, so this is simple although here, but you can certainly get questions like match the columns where the name of the phylum is given on one side and on the other column we have the various levels of organization. Okay, so after this, next, once we know that cells are present or tissues or organs are present, now next is the body plan, that how these all structures they are arranged in the body of the organism. So that would be our next basis of classification, that is the body plan. Okay, so let me take it on this side. So next, that is body plan. Okay, now in this body plan, first one, that is, you can say cell aggregate body plan. Cell aggregate body plan. Now aggregate, the term itself is just a normal English word which means collection, accumulation, right? So here the body comprises a group of cells. The cells are grouped together to form the entire body. And this is also sometimes said as a republic of cells. Like republic, you know that our country is a republic. All of us united make the country, while each person has his or her own individuality, her own freedom. So, but together, united, we are making up the country. So, same way here the cells are grouped together to form the body, but each cell is having some degree of freedom, you can say. So, that is why when porifera will be studying, you will see that the cells can move within the body. Like you will be studying certain cells like amoebocytes, there they are moving within the middle part of the layer, and that way you can see that mobility is certainly present. Okay, so in this case it is, as I said, it is a republic of cells. So here the body is a republic of cells. So here cells have some, like when it is tissue, then this degree of freedom would be lost. Okay, like in epithelial tissue, you see the cells are attached to the basement membrane, so that degree of freedom has been lost. But here such a thing is not there. They are still free to move. So cells have some degree of freedom. Okay, and here the only example with this kind, that is once again the phylum Porifera. Okay, this is Porifera. Then coming to next one, that is blind sac body plan. Blind sac body plan. Now blind sac, why is it so called? Sac, just like any sac in which we carry things. There you will see there is one single opening through which we put the things inside, and through that opening only we take it out, right? So here the body is in such a manner that, like suppose I will give you a diagram here. Suppose this is a cnidarian. So within it, if we are taking a section through the body, this is how we will see. Okay, so there is one single opening, that is mouth, and it is through this mouth that the animal will be taking in the food. That food would be digested, and then the undigested waste will be egested out through the mouth only. Okay, so in blind sac body plan there is, as we said, single opening, there is a single opening which is serving the purpose of both mouth and the anus. Now, which all phyla they have the blind sac body plan? If we see that, so here it is Cnidaria, Ctenophora and Platyhelminthes. Cnidaria, Ctenophora and Platyhelminthes. So these are the three phyla which have the blind sac body plan. So by the time you can take it down, and let me check if anybody has sent any doubts. So once these are in very short, this will help you in quickly revising such wide topic, and that way and basically for solving MCQs, this is what you actually need. Okay. So as we were discussing that the body plan is the body plan where the structure that we have mentioned, that the body may be composed of cells, or it is tissues, or tissues have organized to form organs, and then organ systems are there. But how are they organized in the body? That's what is the body plan, which can be of three types. Two of the types we have already mentioned, that is the cell aggregate body plan, where the body is just a group of cells which have still some freedom with them, and the other one, that is blind sac body plan, which is a very simple kind of body plan where the body has one single opening which is serving the purpose of both mouth as well as anus. So it is ingestion as well as egestion. Ingestion and egestion both will be occurring through the mouth only. Okay, or here you can say through the mouth the movement is what kind? One way or two way? Of course you can see it is two way, because through this the food will be taken in and the undigested waste will be egested through it. So their digestive system can be said as it is two-way. Okay. Now coming to the third one, I hope by this time you must have taken down what has been written on the board. So this side of the board I'll rub so that we can take up that. Okay. So any of these students who have joined in late, so for them let me quickly tell you what we have started with. We have started with animal kingdom, and animal kingdom, it is also called kingdom metazoa, in which we study multicellular organisms which are eukaryotic, they lack a cell wall, and they are heterotrophic, that means they are consumers, right? And in this we are taking up the basis of classification, that is what is the way in which the scientists have been able to decide that which organism should be put in which phylum. So we have certain you can say ways in which we can sort them out into the different groups, right? So in this basis of classification we started with the levels of organization, which can be cellular level, it can be tissue level, organ level, or organ system level. Then next one, of course, that is the body plan. Now body plan is the plan where we will see that these cells and tissues, how they are arranged in the body, right? So there it is the cell aggregate plan, that is just a collection of cells. The other one, blind sac plan. These two we have mentioned. Now coming to the third one, and this one is tube within tube body plan. tube within tube body plan. Now tube within tube body plan is where there is a complete digestive system now formed. Let me make a very simple diagram here. Now suppose this is the digestive tract, and this digestive tract or any digestive tract you see, basically it's a long tube and with both the ends open, right? It could be broader at one point, it could be thinner at one point, but basically it's a long tube. So I have just simplified it, making it a simple with a uniform diameter. And if the body of the organism, we see the body of the organism, body wall that is surrounding it. So just like a worm like structure I have drawn for you here. So here you see this is the body wall. This is the body wall, and this body wall you can imagine this is the outer tube, and this is the alimentary canal. Alimentary canal, or you can simply take it as gut. Alimentary canal or the gut. And here we have two openings, that is mouth, and here is the anus. So in this case you can see that there is two different openings, and as we said, body wall is making the outer tube, the gut is making the inner tube. So tube within tube, that's how you get the term. And in this case the animal will be taking in food through the mouth. As it is passing through the gut, it is getting digested, and undigested waste will be egested out through the anus. So in this case there is one-way traffic. Okay, so here first thing we can mention is that there is complete digestive tract. So when the digestive tract has both mouth and anus present, such a digestive tract is said to be complete. So this is complete digestive tract. This is complete digestive tract. Okay, and so likewise you can say when there was blind sac body plan, we can use the term that here it is incomplete digestive tract. Okay, so this would be complete, while in case of blind sac it is incomplete. Okay, so one thing is here, and then as we said, body wall encloses the digestive tract, as you can see over here. That's what makes it tube within tube. Now tube within tube body plan example you will find in case of, like here you see up till platyhelminthes we have already mentioned. Platyhelminthes has blind sac body plan because suppose it does have a rudimentary kind of digestive system has started, but digestive system, the alimentary canal is there, but it ends bluntly. There is no opening to the anus, so that's why it is considered in the blind sac only. But in case of the phylum coming next to platyhelminthes, that is Aschelminthes. So here Aschelminthes from here to chordata. Aschelminthes to chordata, there is tube within tube body plan. So this is about the various types of body plan that exist in the animal kingdom. So the entire group of animals that you are going to study, we can put them under on the basis of body plan into three categories only, and those three categories are the cell aggregate body plan where you have a single example of poriferans, then blind sac body plan where you have three phyla showing blind sac body plan, those were cnidarians or coelentrates, the ctenophorans and the platyhelminthes. And last but not the least, the tube within tube body plan where complete digestive tract has originated, and in this case there are two separate openings, and this is the phyla, that is Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and finally Chordata. They have tube within tube body plan. Now next basis of classification that I am going to take up is the symmetry. So next we can talk about symmetry. Okay. So let me, by the time this side of the board you must have taken up already. Okay. So here, next basis of classification that we are going to take up is symmetry. Now symmetry, once again, first of all, what is symmetry? Now symmetry would be, first of all, you have to remember that it is only on the basis of external characteristic of the organism. So we are not going to the internal. Like even if we talk about us, so internally there isn't any symmetry, right? Because heart is one and that too slightly towards left. So if we cut the body into equal halves, certainly the inner part of the body will not be showing equal parts. It is only the external part of the body which will be showing equal parts or similar parts, right? So first thing that is to be taken into account is that symmetry is on the basis of external morphology only. And if the animal's body can be cut, that we take a certain plane, by that plane it can be cut into equal halves, the animal is said to be symmetrical. Or you can even talk about any object also. If you see any object and you are able to cut it into equal halves, that means that object is symmetrical. So that is just an analogy by which you can easily understand the concept of symmetry. And if the animal cannot be cut by any plane, there is no plane by which the animal can be cut into equal halves, then the body is said to be asymmetrical. So we can begin from there, right? Poriferans, we had mentioned that mostly, even your NCERT, right, they are mostly asymmetrical. Why is it so? Let us just see through by the help of a diagram. Like suppose this is the substratum, because after all poriferans, you know, are sessile. Okay. So here let us say this is a sponge attached to the substratum, and if you see the sponge, you will find that their body is like the shape of a flower vase. So flower vase like body, and you can very well see that you can cut it into equal halves, is it or not? But normally the sponges will not be just like this. They will have number of branches coming out from it. Okay. So number of branches are arising from different regions going aside like this. I'll just try to make it little three-dimensional. And in such a case, now can you think of any plane which can divide the body into equal halves? No, it cannot be, right? So first thing we can see that poriferans are mostly asymmetrical. Okay, so asymmetry. So here we can mention most poriferans are asymmetrical. As their body cannot be divided into equal halves by any plane. Okay. So this is one. Then next comes about the symmetry. So first thing is there any utility of symmetry? We give so much of importance in zoology to symmetry. So you see, when the organisms started moving, so in that case some level of symmetry was required, because that way the body would be balanced. So all sides of the body would be balanced. You can otherwise very well understand that if the body is asymmetrical, then that means for it to move properly without tumbling here and there, that would be too difficult, right? So that's why some level of symmetry did arise. Although initial symmetry was basically for food gathering, that was radial symmetry, we are just going to take up that, and then more with the motility, it is bilateral symmetry. Okay, so I can take up from this side. I hope you have taken up this side of the topic. So taking up the symmetry. So in the animal kingdom, the symmetry is basically of two types, that is radial symmetry and the other is bilateral symmetry. Radial symmetry and the bilateral symmetry, right? Now radial symmetry is where the body of the organism is in the shape of a cylinder, you can see. Okay, so let us take an example here. Like all of you have at least seen the diagrams of a sea anemone. So number of tentacles they have. So more or less it looks like a flower rather than an animal, but anyway these are the tentacles here, and here the rest of the cylindrical body we can mention, and this cylindrical body, and here in the center there would be mouth. This is attached to the substratum. This is substratum here. Now in this case you have to see a longitudinal axis. So there are few things, because radial symmetry normally you think that there are, all of you I hope know that there are many planes which can divide the body into equal halves. But here you have to first consider that what is the direction of that plane. So the direction of that plane is longitudinal. That means going like this, going through the center from the oral to the aboral side. So longitudinal plane we have to consider. And this longitudinal plane, how many can be there which can divide the body into equal halves? So of course here, as you can see, since it's a cylinder, so you have to just keep this shifting 360 degree, you can keep on rotating it. So that means infinite planes are there which can cut the body into equal halves. Okay, so radial symmetry is that body can be cut into equal halves by infinite, but at the same time I'll add on longitudinal, infinite longitudinal planes. So the body can be cut into equal halves by infinite number of longitudinal planes, right? So in such animals, the side where the mouth is present, that is called oral side, and the side which is towards the substratum, that is called aboral side. So the oral side and the aboral side, these are the terms that are used. Oral word comes for the mouth. So where the mouth is present, that is the oral side, and where there is the side which is away from it, that would be the aboral side. Okay, and such type of symmetry, radial symmetry, is suitable for organisms which are sessile. Okay, which are sessile, but at the same time they are catching their food. Because after all, poriferans are also sessile, but with poriferans, I hope all of you know that the water current is continuously passing through the body, and along with that water current they are filtering some microscopic planktons, organisms on which they can feed on. So they are basically filter feeders. But where the organism is sessile, but at the same time it is a carnivore as well. So these cnidarians, you will see they are carnivores. So they have to catch their food, and in this case, since the body is in such a way that it is alike on all the sides, so it can gather food from any side, from the water surrounding the which is surrounding the animal, from any side they can catch their food. Okay, So this thing we can mention, of course, it is suitable for sessile animals. Sessile means which are attached to the substratum, which do not move. So it is suitable for sessile animals. Okay, that is there. And where do we find radial symmetry? The example next we can take is, the example here is after porifera, next one, that is Coelenterata, Ctenophora, and there is one very strange group, that is Echinodermata. So here it is adult echinoderms. Okay, so just take this much, then we will go on with bilateral. Just hello. Okay, so we have few doubts over here. Okay, right. So Vidushi has asked a doubt, that is regarding blind sac body plan. Okay, blind sac body plan, and this she has not been able to understand, doesn't matter. So I think I can clear this side of the board. I will first take up that doubt and then we will move on to the bilateral symmetry. So here now you see blind sac. What blind sac, why the term is used? Just try to imagine a bag in which you put your things. Okay, so there is one single opening to put the things, and when you have to take it out, you have to take it out from here. You don't have a zipper here. Okay, so if there was a zipper here, let us say, so you could open this side and you can take out the things from here also. So but it's not so. Here the body plan is in such a way, that there is one single opening, and that's what I was trying to show you in the diagram, that the diagram we had taken in case of Hydra. Try to imagine Hydra. Now this Hydra, you will see that it is having a mouth, the tentacles all around it. I am just drawing two, but there would be 6 to 10 tentacles are there, and inside here you will find that this is a section we have taken. So outer wall is their epidermis. Okay, you needn't label it at the moment, but anyway, the inner lining is the gastrodermis, and this cavity here, this is called their coelentron. That's the reason this particular group has been named as Coelenterata. Okay, just a sec, I am coming to that. Divyam, I'll just take Divya, I'll take your doubt as well. So this is coelentron. Now in this case, they catch the food by the help of their tentacles. Their tentacles are outside, they'll catch the food, and this food they are going to put through the mouth into this coelentron. Now the food is going to be digested, and after digestion the waste which are left out or any undigested part which is left out, that will be egested out. That means it will be sent out of the body through the mouth again. So there is one single opening through which they are taking in the food and through which they are egesting out their feces. So this is one single opening that we need, that we come to know about, right? So such type of body plan is blind sac. This is blind sac, whereby and how you can distinguish it, that is why the other one that I took, the tube within tube. Now tube within tube, try to imagine your own digestive tract, isn't it? So you see, starting from the mouth, then coming to the stomach, intestine, and finally opening to the outside through the anus, and this is all enclosed inside our body wall. So the body wall which is outside, that is making the outer tube, and the digestive tract is making the inner tube. So that becomes tube within tube, where there are two separate openings, mouth and anus. Mouth and anus, mouth for ingestion, taking in of food, and undigested waste, that is the feces, will be egested through the anus. So I think this must be clear now, Vidushi. And another question Sujata has put, that poriferan body can be divided into vertical. So vertical division is possible, certainly, but that position once again is only when there is no branching. As I told you, it is like a flower vase, and if that flower vase, certainly you can cut it into two equal halves, but there are so many branches coming out from different directions, so that's why it will be more or less asymmetry. And Divya has asked that how many openings are there in the sea anemone. So in the sea anemone, you can see there is one single opening, that is mouth. So this is also a cnidarian, and cnidarians have blind sac body plan, so there will be only one opening. And Kumar has asked that sessile animals, sessile means which do not move. So sessile means, like here I have shown, it is attached to the rock. It is attached to the rock, it is not going to move about here and there in the ocean. So such animal is said to be sessile. Okay, so as I told you, that radial symmetry is more suitable for animals which are sessile, because with radial symmetry, what is their body from all sides is similar. So from any side they can gather the food. Okay, and Ashwarya, there is a question, what about planaria? So planaria comes under platyhelminthes, and in platyhelminthes bilateral symmetry starts, but the body plan is still blind sac. It is blind sac because in platyhelminthes the digestive tract is incomplete. Right? That means it starts from the mouth, but then it ends abruptly. That means the lower end is not open. It's not open into, there is no anus. Anus is absent. So mouth will be for ingestion as well as egestion. Okay, so I think I hope your doubts are cleared. So we can take up the bilateral symmetry now. So this side I'll rub off. Okay. So here, next thing, coming to bilateral symmetry. Now bilateral symmetry, you will see that it came with when the organisms became motile. Corals are these only, coelentrates which are belonging to one particular class of coelentrata. They are called the corals, and in some of them, usually the term coral we use when their body is having some calcareous deposition, and because of that calcium deposition it's very hard, so they have a skeleton is formed which is hard, and those are the corals. Okay, so corals, aboral side is just the term, it's a term for the side which is opposite to the oral. Oral is the side where mouth is present, So here you see oral side, this is the side where mouth is present, mouth here you can see, and the side which is opposite to the mouth, so that is the aboral side. Okay, I think that should be clear now. Okay, so now coming to the bilateral symmetry. Now bilateral symmetry, in this case bilateral, that means two lateral sides are there. Okay, so in this case you will see, like we can draw a simple cartoony animal so that it becomes more easy to understand. And let us say, let us put the appendages also, hands and feet, or I think that will be easy to understand. Now in this case, try to think how many planes are possible which can cut the body into equal halves. So here if you cut like this, can you get two equal halves? No, you cannot, right? So the only way out is cutting in this manner, cutting like this. So in this case, the two sides, these two sides are the lateral sides, and there is only one such plane possible. So you see, in case of radial symmetry, we started with infinite number of planes, but here the number of planes are just one, only one, and that plane which is going through the center like this is called the mid sagittal plane. So it's a longitudinal plane which is passing through the body, cutting it into two halves, and these two halves will be mirror halves, the right half and the left half, isn't it? So these are mirror half, like in the mirror when you put an image, so the right side in the image appears to be left, isn't it? So same way the left and the right side will be there. So in this case we can mention, I'll mention it here for bilateral symmetry. Okay, so single mid sagittal plane Now mid sagittal once again is a longitudinal plane, but all longitudinal planes, like longitudinal plane you can even cut from this that way also, but this is not cutting the body into equal halves. So the plane which is the longitudinal plane which is passing through the center of the body, then that would be mid sagittal plane. So that makes it very clear that from where we are cutting the body. Okay, so a single mid sagittal plane divides the body into right and left mirror halves. Now this symmetry is suitable for where the animals became motile, right? Try to think that since there are two lateral sides, like suppose when we are walking, so we move our hands also, we move our feet, or when you go to swim, so then also you are using both your hands. So both sides when they are equalized, then only there would be proper balance for the movement. Okay, so this is suitable for motility. So when the animals became motile, with that there was origin of bilateral symmetry. Now with bilateral symmetry, try to imagine something like this, that again I am making a substratum, let us say in the ocean, and here there is our animal which initially, let us say, it started crawling over the surface and it started moving. Now when it is crawling, so the side, let me make a mouth here. So the side where the mouth is, which is when it is crawling, it will be moving in this direction. So the side which is ahead, that is called the anterior side. That is the anterior. Now we come to this word anterior. Anterior, that means when the animal moves, so the side which is in front, that would be called the anterior side. And the side which is trailing behind, that is called posterior. That is the posterior end. Then the belly side, when it is crawling, so the belly side is towards the substratum, so that is called the ventral side, and the back of the animal, that is called the dorsal side. So now you see, when the animals became motile, they were bilaterally symmetrical, and there was antero-posterior axis and dorso-ventral axis. These were established. And one another very important thing started with it. That was that when the animal is moving, the mouth is this side, and with the mouth they have to catch the food, right? So most of these sensory structures slowly started accumulating near the mouth. And near the mouth, when these sensory structures started accumulating, that led to the development of the head and the brain. Okay, so that's why we use a term here, cephalization. Now cephalic word comes for head, and what is inside the head, if it is there, so that is encephalon, that is brain, right? So cephalization began with the bilateral symmetry, and bilateral symmetry began with motility. So you see how many things we can relate over here. So we can list them out. That first thing I have already mentioned, that it is suitable for motility. Then what else happened with bilateral symmetry? That antero-posterior and dorso-ventral axis were established. Okay. So antero-posterior and dorso-ventral axis were established. Okay, and last but not the least, cephalization. So now you get a word here, cephalization. Cephalization is formation of head, because as I told you, cephalic word comes for head. Okay, so cephalic, this word means head. So cephalization begins. And on a lighter note, one very important thing you can remember here, that since cephalization began with bilateral symmetry, their brain also exists only in bilaterally symmetrical animals. So the animals which are radially symmetrical, they don't have a brain. So you can note it down. This is a very important point, that brain is absent in radially symmetrical animals. All the sides of the body are similar, so there is nothing like brain over there. Okay, so you can imagine the sea anemone that you were asking just now, or the jellyfish, they don't have any brain, so they have been existing without brain. Yes, there is a Cheyenne, I think this is the name, cephalization. So cephalization is the formation of head, formation of head, and I told you why this head formed, because when the animals started moving, so initially you see they didn't have any limbs initially, so they must have been just crawlers. They crawled over the surface of the ocean, means the rocks which are inside the ocean, and when they were crawling, the side which was ahead, that was the side where the mouth was there, and the mouth was meant to catch food. That was the only purpose. And so to sense their food, what happened? All the sensory structures started moving, or means you can see in the development occurred in such a way that all sensory structures, they were near the mouth, they arranged near the mouth. And when this all happened, so many nerve cells, you see they are coming near, means they are accumulating at one point. So what's that is happening? That is the development of the brain also. So with the head, there was evolution of brain as well. Okay, so this is what was there, you see. And even in our case, you can see where the head is, within the head there is brain. And don't you think our most important sensory structures are also located here? The eyes are here, the ear, nose, the tongue with the taste buds in it, isn't it? So you can see most of the important sensory structures, they are in this region only. So why do we have that? Because in the course of evolution, this is how it has evolved, isn't it? It would have mattered to us if you think that instead of having the eyes over here, we could have had it at the back, or maybe ears at some other location of the body. So why all these important structures are there in the head region only? So the reason behind it, that's what I have told you. Okay? Yes, Aishwarya, certainly. Humans do have the process of cephalization, and in the development we can see that when we study the embryonic development, so that is a very important part of the development. And if in any embryo for any deformity there is, cephalization doesn't occur, that embryo will not complete the development also. And okay, Pankaj, yes, I am just going to tell you all animals which have bilateral symmetry. I am just coming to that. And radially symmetrical animals, Vinayak, don't have a brain, so how their body function happens? So they do have nerve cells. You see, they have nerve cells. The coelentrates, you will see they have nerve cells, but the nerve cells are you can say diffused pattern is there. That means they are distributed all over the body, spread out all over the body, but they are not collecting together to form a brain. And this used to be an old question. In the earlier times in the medical entrance exam, there was a very common question, many a times it appeared in the various exams, that select the animal out of the following which has a nerve net but no brain. So the nerve cells are spread out over the body forming a network, but they are not collected together. Like in our brain, you know, that we have billions of neurons collected together, so they are not collected together to form a brain. So that was out of the animals, usually Hydra was there, and Hydra, you very well know, is in the Coelentrata group. Okay, so let us put the examples here, radially symmetrical. And one more thing, once again on a lighter note, I will tell you, Vinayak, that since you have said that they don't have a brain, it is us actually, we give so much of importance to our brain, we cannot do anything about the brain. But these animals, they are successfully living. Rather you would say that jellyfish, or means any of this coelentrates, these jellyfish have been surviving way back 500 million years ago. From there they originated, and they are surviving till date. And if you have in the 12th you have studied human evolution, so you will see that modern humans, they have just appeared about 75,000 years ago or so, and in that time, that little time, we have brought the earth to the verge of destruction. So I think without brain it would have been better. Anyway, so here taking the examples of radially symmetrical and bilaterally symmetrical animals. So in this case, I can rub off the diagrams, I think, and I can put over here. So radially symmetrical, we have already mentioned, Coelentrata, Ctenophora, and adult echinoderms. Mind it, because their larva is very much bilateral. Okay, so here remaining phyla which we are left out with, that is, the examples are, examples here, Platyhelminthes, okay, Platyhelminthes, starting from there going up to chordata, okay, Platyhelminthes to chordata. So in the beginning of the class, even if some people were not there, we can list out the entire phyla. Platyhelminthes, after Platyhelminthes you have Aschelminthes, then Annelida, Arthropoda, Mollusca, Mollusca, then Echinodermata. Now Echinodermata, larval Echinodermata, okay, larval Echinodermata that has bilateral symmetry, and then Hemichordata, and finally of course chordata, right? So from Platyhelminthes to chordata, we can certainly list it out like that. And if you Okay, yes, Aryan, if Hydra undergoes budding, so Hydra, when it undergoes budding, so bud arises from the parent body and then it detaches. Of course, during that period, you can certainly say that symmetry, radial symmetry cannot be observed for some time, certainly. But this is a generalized thing, because budding is, of course, done by once the animal has become adult. Otherwise, normally, the bud will always not be there, right? So that way we take it as in this manner. Okay, so platyhelminthes to chordata, this is bilateral symmetry. So that's why in some of the books you will find they have even a classification, that is the entire eumetazoa. Eumetazoa, I told you, they are the true metazoans, true animals where there is tissue, organ system, germ layers, and also the symmetry present. So that eumetazoans are classified into radiata and bilateria. This also is one way of classification, but since it's not there in the NCERT, we can skip it, right? So next basis of classification we will take up, and that next basis is, I'll talk about germ layers now. So because we have talked of levels of organization, I hope everybody had joined in by that time. So levels of organization, we have mentioned 4 of them: the cellular level, the tissue level, organ level, and the organ system level. And then we talked about body plan, which were of 3 types, that is cell aggregate body plan, blind sac body plan, and tube within tube body plan. And next we have taken up the symmetry. Porifera we have separated out because porifera are asymmetrical, mostly asymmetrical, and this mostly is also a very important term you see in your entrance exam, because biology, you have to remember, biology is a science of exceptions. So if in any question by chance you get a statement written like that all poriferans are asymmetrical, so there and then itself that would be a false statement, that would be wrong. So that wherever the statement starts with all or it ends with only, it should always ring a bell. So you should know that this cannot be correct because there has to be an exception. And mostly in most of the phyla we'll see some or the other exceptions are there. And if in some of them exception is not there, that itself becomes an exception, right? So anyway, we'll move on to our next thing, that is germ layers. Okay. So next basis of classification after symmetry. I hope you have taken down the examples. Platyhelminthes to chordata, they show bilateral symmetry, and this side we had already done. Okay. So here you see next thing, that is, I think this would be our 4th one. You can keep the counting. So next one, that is germ layers. Now germ layers, you see, germ layers comes from the term germinate. So just like in a seed, just a sec. Very right, Tushar, very good question. As adult echinodermata are radially symmetrical, do they exhibit cephalization or is it lost from larval to adult stage? Very valid question, very good to bring up this question. Yes, the adult echinoderms lack cephalization. They don't have a brain. The starfish, the sea stars or sea cucumbers, since in adult life they are radially symmetrical, so they also just have a diffuse type of nervous system. That means the neurons are distributed all over the body, but they are not aggregating together to form any brain. Okay, so the adult echinoderms are lacking that. Coelomates I'll be just doing with you, don't worry, I'll come to that. Let me first take germ layers, because after germ layer that becomes more easily relatable. So this is the sequence I prefer to follow. Okay. So anyway, coming to germ layers. Now you see germ layers, as I told you, they come from the term germinate, because when the embryo is developing, so this embryo initially, I'll start rather with a diagram that it becomes more easy. Every organism starts its life from a single cell, and that single cell is zygote, right? Now this zygote starts with cleavage divisions. It starts with cleavage divisions forming 2-celled stage, a 4-celled stage, so on. This you must have studied with the human reproduction chapter in 12th, whereby you study the human development. So this is how it is going to develop into, and then from here it forms a solid ball of cells, which is called, okay, so, and that is morula. Since you have studied for humans, so I'll take up that way only. Morula, this is solid ball of cells. Okay, and this morula, which is solid ball, this then transforms the cells, rearrange so that it forms a hollow ball of cells. So I'll put the arrow here. It forms a hollow ball. So this is in section, of course, we are seeing. From outside we won't be able to make out whether it is hollow or solid, and a hollow ball of cells, which is called blastula. It is only in mammals that we use the term blastocyst. Since here I am taking Animalia in general, so I'll use the term blastula, and when you will be talking about mammals including humans, then you will use the term blastocyst. Okay, so there shouldn't be any confusion there. So blastula, this is hollow ball of cells. This is the hollow ball, and here there is a cavity now. This cavity is the blastocele. So the cavity, cele word comes for cavity, and since there is a cavity within the blastula, this is called blastocele. Now what happens next stage, that is after this cavity has been formed, the blastula has been formed, these cells, they start rearranging themselves. There are a lot of movements, cellular movements taking place at this time, so that it transforms into next layer, next stage. I'll rather put it here so that you can see. And you see some of the cells, they have started moving in. Now the cells which have started moving in, as a result, what has happened? There is a cavity which is formed inside. Okay, it is formed inside. Now this stage is called the gastrula. This is the gastrula. Okay, this is the gastrula, and in this gastrula you see what has happened. From where it has started moving in, it would be just like a dent. Try to imagine a soft ball in your hand, and in that soft ball you poke your finger. So where you are poking your finger, there will be a small dent arising. That's what we will be able to see, and from that dent, if you keep poking your finger towards the inside, so it is only the outer layer which is going in, right? It is invaginating in. And so from where this dent has started, that would be called blastopore. That is blastopore, through which it is moving in. And this new cavity which has come up, let me rub the other side. I think by this time everybody has taken this. Okay. So this new cavity which is arising, this is called gastrocele, because this is now the cavity of gastrula. This is the cavity of gastrula, gastrocele, or you come across this term also. I think some of you must have certainly studied archenteron. Okay, you see arche comes for primitive, and enteron word or enteric word always comes for the gut, the digestive tract. So this is the beginning of the digestive tract which is being formed over here. Okay, and this cavity that is between the archenteron and the outer layer, this is still the same, that is blastocele. This is the blastocele, but you will see blastocele is slowly becoming reduced. And now what is happening, that the cells which are outside, they would be forming the ectoderm. The one which has come in, that is which is forming the archenteron, the lining of the archenteron, this is going to become the endoderm. So it is in this gastrula stage that these germ layers are established. So the germ layers, the ectoderm, mesoderm and endoderm, these germ layers, first thing we can mention is that they arise during gastrulation. and there was a question. I will solve that, and then I'll move ahead. Jellyfish is immortal, some of them, not all. There are some of the jellyfish, because as soon as there is going to be they become older, so they immediately they reproduce in such a way that they will return to their juvenile form. Juvenile form is what would you say, young form, more young form. They will return to that. They have, some of them have that ability. And difference between protostomic and deuterostomic, I haven't taken up, Ashish, as yet. I'll be telling you that along with this, okay, because I will relate here. Okay, so anyway, as you see, first thing we can mention, germ layers. So germ layers are established during gastrulation. Okay, so first thing we can mention over here is that germ layers are established during gastrulation. Germ layers are established during gastrulation. Okay, first thing which we have seen just now. And how many germ layers are going to be formed? On that basis, there can be only two types, either 2 or 3. So accordingly, we have two terms here, that is diploblastic animals. Now diploblastic is 2 germ layers. 2 germ layers are formed. 2 germ layers are formed. When are they formed? In the embryonic life. And where in the embryonic life? In the gastrulation phase. Okay, 2 germ layers are formed, and these 2 germ layers are ectoderm and endoderm. So just now I mentioned with you that the outer one, that will become the ectoderm, and the one which has come in, which is lining the gastrocele, or you can say which is lining the archenteron, that is supposed to become the endoderm. Okay, so 2 germ layers are formed, ectoderm and the endoderm, and in between these 2 layers there would be a jelly-like substance filled in, which will not be having cells in it, that is called the mesoglia. Okay, so there is non-cellular mesoglia present in between. Okay, so for a diploblastic animal, as your NCERT has given, you can get a diagrammatic question on it. If you take a section through it, if you take a section, so like once again our favorite example, the hydra. Let us cut through this hydra once again. Okay, so if we take a TS through this hydra, we cut through it. Okay, you take a TS, transfer section. So what you will find that here, just now I was telling you that on the inner side there is going to be the endoderm, which here becomes the gastrodermis. So here you have this cellular layer. This is the cellular layer, and here this is the ectoderm. Okay, so here ectoderm, this one is the endoderm. I hope up till here it goes, you can see it, and in between you will see, in between there is mesoglia, a gelatinous layer, that is mesoglia, and this is of course their body cavity, this one inside, where the digestion occurs, the coelentron we had mentioned about. Okay, so this one is the coelentron. Okay, so this kind of arrangement, when you see, this is a diploblastic animal. So in this, I think the this side I can rub off, and I'll just put it here. Diploblastic animals, the example you have, diploblastic animals, The example is Coelenterata and Ctenophora. Only two. Okay, only two phyla are there which are diploblastic. Okay, which are diploblastic. So one more thing you can relate here, one very interesting thing once again. If you remember, Coelenterata and Ctenophora, both of these were also radially symmetrical. Okay, so leave out Echinodermata, because Echinodermata is a strange case where it is one part of the life cycle it is bilaterally symmetrical and the other part it is radially symmetrical. So leave that. So truly which are radially symmetrical all through their life, those are these two phyla, and both of them are diploblastic. So we can link these two things together, that these two are also radially symmetrical. Okay. So we are almost running out of time. So triploblastic animals, I think I will take up in the next session tomorrow, and I will because many of the things I have taken up with the embryology, I think some people might have a small doubts related to that. So I will certainly take up that as well once again, and by the time we can quickly recap whatever we had done today. So as I told you, we started with Animalia, which is also called metazoa, and metazoans are multicellular eukaryotes lacking a cell wall. They are heterotrophic. They have the ability for locomotion at least in some stage of life. Even if the adult is sessile, you will find the larva is very much motile, and they have the ability to sense their surroundings. So some kind of sensory ability they have. Okay, and then we took up the basis of classification, in which we have talked of levels of organization, where we have taken 4 levels. Then after that the body plan, that is cell aggregate, blind sac and tube within tube. Then I have talked about symmetry, and I have related number of things with the symmetry. You will see that radially symmetrical animals, we have related that it is suitable, they are usually so sessile. With them radial symmetry is good enough for them because they can gather food from all directions. And once the animals gained motility, with them bilaterally symmetrical condition was more suitable, and with bilateral symmetry there was also beginning of cephalization, that is the beginning of the head and the brain. Okay, and then of course we had just started with the germ layers. Germ layers, as I mentioned with you, they develop during the embryonic stage, and that is during the gastrulation phase. And these germ layers, depending on whether they are 2 or 3, the animals can be diploblastic or triploblastic. So triploblastic certainly we will be taking up in the next class, that is tomorrow. And with that, certainly Ashish, your question will also be solved, that is protostomic and deuterostomic condition, because that one I take up with that. Okay, so don't think that your question has not been answered, it will be certainly taken up. So I think we can call it a day. You have sat through the entire class, and I'm delighted to have you all here, and there have been some really nice doubts being raised. So good to go. So we can call it a day. Thanks for being here.